on Friday from 9 to 10. So you have plenty of time to come in to look at your exam. So you don't have to email me. Just come in during those times, and you're more than welcome to look at your exam. Anyway, so that's that, right? So it's not just my office hours. So if you have class during my office hours, I understand. But there are other times you can come in as well. And it's in Sequoia 120A as in Apple, not 128. Again, all of this information is actually on the syllabus. Again, I'm not sure why, I know I have a strong Texas accent, but I'm not sure why this information is not clear. Okay, that's okay. All right, so how am I doing? Is everybody okay so far? You're still intact? We're going to go through 4 or 5 major pathways today. They're pretty intense, they're pretty detailed. But as with everything, these are just examples. So these are detailed examples. What you should be able to do is draw this and should be able to make more general conclusions. And as you have seen on my exams, a lot of questions I ask are more general based on the examples that I give, right? So for instance, when I ask about, we'll talk about phosphorylation to turn on or turn off certain enzyme. On the exam, I will ask about phosphorylation in general. Would phosphorylation activate or inactivate a protein? And your answer should be, it doesn't always activate or inactivate, it should regulate protein function, right? That's how this works. So these are, and in talking about the specific details, for instance, you can see that we'll start out with a G protein link receptor, right? And you will see that some receptors can actually activate multiple pathways. So in talking about these details, we'll talk about bigger processes, bigger ideas. For instance, divergent crosstalk. That's what that means when you have one receptor activating multiple pathways. Or sometimes a particular enzyme may be activated by different pathways, and we'll call that convergent crosstalk. So again, from these details, I will tell you more bigger ideas. And again, really the bigger ideas is what you should really pay attention to, right? And as you notice on the exam addresses mainly bigger ideas, okay? So we'll go through all of these pathways, including talking a little bit about plants. G protein link receptor. So I said that G protein link receptor, G proteins in general, we already talked about G proteins, what that is. So G protein link receptor is a receptor, transmembrane receptor, that is linked to a G protein. So the receptor itself is not a G protein, but it is linked to a receptor. So this is a G protein link receptor with a signaling molecule binding site on the non-cytosolic side. The cytosolic domain has a binding site for a G protein. Okay? So G proteins are categorized or divided into 2 major superfamily, because both of them are pretty big. One is called trimeric G protein, and the other one is monomeric G protein. We've already seen monomeric G proteins. For instance, diamine, tubulin, those are, remember all of the proteins that have TP bound? They are G proteins, right? G protein or GTPases. So those are monomeric. These are trimeric G proteins, okay? And G protein link receptors, only trimeric G proteins are linked to a receptor. Okay? So whenever we talk about, again, G protein link receptor, we're really talking about the trimeric form. We'll talk about the monomeric form very shortly today. But again, any G protein that is linked to a receptor is a trimeric G protein. Interesting thing about G protein link receptors is that if you look at all of the legal ones, the legal pharmaceutical drugs, those of you who want to go into the pharmaceutical area, about half, more than half of these these drugs, guess what? They they target G protein link receptors. That's how prevalent and how important G protein link receptors are. Okay, so that's why we're talking about them. So so trimeric, because there are 3 different subunits, you're not required to know which subunit does what. So, but basically, for the trimeric G protein, here's how it works. Okay, one of the subunits, and doesn't matter whether it's the alpha, beta, or gamma. One of the subunits actually contains the catalytic site. Right, and the other 2 units. And then one of the other 2 units actually contains the GDP or GTP binding site. So far so good. And then there's also one of the subunits also interacts with the receptor. So you have 3 subunits. Each one has a slightly different function, basically. Right? Okay. So the enzymatic activity that causes the exchange of GDP for GTP is inherent, is intrinsic in the trimeric G protein. This is important because when we talk about the monomeric G protein, that enzymatic activity belongs somewhere else. So there has to be an additional enzyme that catalyzes the exchange. But for a trimeric G protein, and this is the point. The point is that for a trimeric G protein, that there is an intrinsic enzymatic activity that can allow for the exchange of GDP for GTP. And when is that intrinsic activity activated? So we'll talk about all of this. When is it activated? Here's how it works. Normally, the trimeric G protein has a GDP bound to it, so therefore it's inactive. In order to activate that, this is what happens is it is linked to an inactive receptor. When there's a signal that comes in and activates the receptor, right, that binds to the receptor, thereby activating it. Activating means that there's a conformational change in the receptor. That in turn would cause a conformational change in the G protein, which therefore activate the intrinsic exchange activity that causes that exchange. So again, you don't need an extra enzyme that catalyzes the exchange. So in order again to activate that G protein, then the signal will activate the receptor, which in turn activate the intrinsic enzymatic activity that catalyzes that exchange, right? So that the exchange occurs. When the exchange had occurred, now the whole G protein is now active to carry out yet another function. So you see the trimeric G protein has 2 different activities, right? One activity is the exchange of the GDP for the GTP, so that it can activate itself. And then once active, then the G protein can activate other proteins. Does that make sense? So it's important for me to explain that because when we talk about a monomeric G protein, the monomeric G protein does not have this intrinsic enzymatic activity to exchange GDP for GTP, but when a GTP bound, it still has, so therefore a monomeric G protein only has really one enzymatic activity, and the trimeric G protein has actually 2 enzymatic activities, right? One of which is an intrinsic activity to exchange GDP for GTP. I must have said that about 5 different ways and 5 different times. Is it clear? Is the point. I can talk until I'm blue in the face, but if it's not clear, how else can I explain it? We're good? Yes, sir.
The transmembrane protein part, is that just the receptor
right.
of the trans?
Right, so this is a G protein linked receptor. So this is a receptor, it's linked to the G protein. It's always interacting with the G protein, but it's so that's hardwired, right? And then when the receptor is activated, the receptor in turn activate the G protein, activate the intrinsic exchange GDP for GTP exchange activity, right? Only then after the exchange, then the G protein itself has yet another activity, enzymatic activity, to carry out some other processes.
Is it cleaved? It looks to be cleaved.
Yeah, don't worry about, yeah, that's, a lot is going on here, but don't worry about that either. Yes, sir?
For the 3 subunits, you said one contains the GDP binding site, one interacts with the receptor, and what is the third one?
So here are the things, right? So you have, in order for the G protein to link to the receptor, it has to have a binding site for the receptor. And then in order for the GDP to exchange for the GTP, it has to have that enzymatic activity. And then when the GTP is bound, it has yet another catalytic activity. That's it. Does that make sense? I need you
Yeah,
to.
so it's the third one's the enzyme binding site.
Right.
Basically, one for the
The
receptor,
enzymatic,
one for
right.
the GDP, one for the enzyme. Okay.
Okay. And then hydrolysis of the GTP, right, would inactivate the G protein. Okay. GTPase, should I explain that? No, don't worry about it. GTPase has a specific meaning, but I'm not going to go into that. So just know that hydrolysis of GTP then would inactivate the G protein. That's enough. Okay. I'm debating with myself. Once a G protein is active, so this is when it has a GTP bound. A G protein can activate multiple pathways. Right. We'll look at the pathway, the cyclic AMP pathway versus the phospholipase C pathway. Okay, so we consider this to be a divergent crosstalk because one enzyme, one part of a pathway that can diverge and activate multiple enzymes or activities. So we refer to this as divergent crosstalk. Okay?
Diversion?
Divergent, right. Diverge, right. Divergent crosstalk. Okay? So let's look at how the G protein actually can activate the cyclic AMP pathway, and what is a cyclic AMP pathway, and what is really the effector function is the important part, right? So when activated, this G protein then can activate what we call effector enzymes. So in order to activate the cyclic AMP pathway, then the effector enzyme for the cyclic AMP pathway is adenylyl cyclase. Remember what effector means? Effector just means at any particular step, that's the goal, right? That's the goal for the activity. So the effector enzyme after the G protein is activated, in this case, is adenylyl cyclase. Adenylyl cyclase, when activated, will convert ATP into a cyclic AMP. So this is why this is called a cyclic AMP pathway. Right? So cyclic AMP belong to a group of molecules. Some of them are organic molecules, some of them are ions. Belong to a group of molecules called a secondary messenger. Okay? So calcium ions are secondary messengers. Later on we'll talk about IP3 or inositol triphosphate. But so cyclic AMP is a type of second messenger. If it's a second messenger, what's the first messenger? The original signal, right? The original signaling molecule. So again, so that's its first messenger, passes down the information onto the effector enzyme here. The effector enzyme then would catalyze the conversion of ATP to cyclic AMP. Okay, cyclic AMP then can be used as a coenzyme for a number of enzymes, including an enzyme called protein kinase A. So it's a kinase. Okay, PKA or protein kinase A. Protein kinase A normally is in an inactive form that it's a tetramer, right? Two of the subunits contain binding site for cyclic AMP, and the other two subunits are catalytic subunits. When the catalytic subunits are bound by the regulatory subunits, they are inactive. So the idea is to allow the regulatory subunits to release the catalytic subunit. How do you get the regulatory subunits to release the catalytic subunits? By the binding of cyclic AMP. How do you think that happens? What happens when the catalytic cyclic AMP binds to the regulatory subunit that causes the release?
It's conformational change.
How about that? Thank you for saying that. I would pull my hair out if the little hair I have if you did that. So then this all happens in the cytosol. pKa just happens to be a regulator of transcription factors. And transcription factors are in the nucleoplasm. So what happens is the pKa then will be transported into the nucleoplasm along the microtubule using diamine as a molecular motor, right? All of that stuff through the nuclear pore, okay? Just like that.
Oh, okay.
pKa then, the catalytic subunits then would phosphorylate a number of transcription factors. In this case, the example would be CREB. Don't call it Krebs because that you will confuse with the Krebs cycle, right? Which is KREB. So it would normally Krebs in the unphosphorylated form would be an inactive transcription factor. When phosphorylated by pKa, then Krebs is active and then therefore can bind to a response element to regulate gene expression. In this case, it actually turns it on. So in this case, phosphorylation, both phosphorylation and the binding of the transcription factor will activate gene expression. But again, I say regulate because that's not always the case, right? So that's how the cyclic AMP pathway is activated by a G protein link receptor. So that's what the targeting, the signaling cell wants a target cell to do, is to carry out gene expression of this particular gene. Let's say the signaling cell wants a target cell to divide. Then this gene, the target gene, would be a gene that is involved in cell division. Okay, for instance, making more tubulin dimers for spindle fiber formation, for instance. Is that? Are we good with all that? Yes, sir. Wesley.
Was the pKa itself a transcription factor?
No.
Or just activates?
It's a kinase.
Okay, yeah.
Yeah, so it activates the transcription factor, which is the CREB.
Okay.
Right. And so what are some of the bigger ideas about this? Some of the bigger idea is the fact that you do have a signal transduction pathway. At this point right here, here's where amplification occurs. Right? So adenylyl cyclase is going to convert a bunch of ATP into cyclic AMP. Right? And it's only turned off when GTP is turned off, and the G protein is turned off. And the G protein is only turned off when the GTP is hydrolyzed. So how do you control the amount of cyclic AMP that is going to be produced? By controlling how long, the longevity of the active G protein. You see how that works? So that's the bigger picture, is to think about that. Yes, ma'am?
Sorry, can you repeat that a little bit slower?
Yes. So one of the big picture is the fact that right here, here's where amplification occurs, right? Amplification occurs. So you're not going to produce just one cyclic AMP. You're going to produce a bunch of cyclic AMP, right? And how is ATP converted to a cyclic AMP? By the active adenylyl cyclase. So far, so good? So I'm working backward here. So the longer the adenylyl cyclase remains active, the more cyclic AMP will be produced. So the bigger the amplification. Good? All right. And then how do you regulate the longevity of the adenylyl cyclase activity? By keeping the G protein active longer. How is a G protein kept active longer? by having the GTP bound longer, or in other words, by preventing hydrolysis of GTP. There is an enzyme that actually catalyzes the hydrolysis of the GTP, or there's a hydrolysis activity, and actually that's also intrinsic in the GTP, trimeric GTPase. So then when the GTP is hydrolyzed, then the G protein is turned off, which in turn would turn off the adenylyl cyclase, which then cut off the synthesis production of cyclic AMP. So to regulate how much amplification you would have, you actually have to go all the way back and regulate how long the G protein needs to remain active. Right? That's the big picture. Yes, ma'am. There's no more, and there's no. One thing is not more important than the other. They're all important because any one of these that's missing, then you're not going to carry out gene expression. Yes, ma'am. The signal, you're talking about cell-specific, yes. We'll talk about specificity in a minute. Right now, we're just in general, right? But then this is just a ubiquitous receptor. This is just a ubiquitous G protein, which is not the case, right? There are specific ones, and we'll talk about that at the very end to summarize everything. But that's a good question. Yes, ma'am.
If you're producing a lot of cyclic AMP and activating many PKA, so all of the PKAs, are they going to all target the same area of the gene to produce multiple transcripts? Or?
Okay, so yes, to everything. So cyclic AMP can be used as a coenzyme for a number of different enzymes, not just PKA. So it's just PKA that we focus on, but it could be a number of different enzymes, right? Number one. Number two, then PKA can actually phosphorylate a number of different transcription factors. Right? And then each transcription factor may bind to a number of different response elements of a number of different genes. All of that depends on what? Gene expression depends on whether or not you have the gene actively inactive, whether or not you have methylation, everything that we talked about, right? So this is just a summarized version, but if you're going to look at it in a lot more detail, what Kelsey was asking, so the receptor is cell-specific, so not all, so the signal is cell-specific that activates a specific receptor that activates a specific G protein. So even when we talk about divergent crosstalk, where a G protein can activate either a cyclic AMP pathway or the other PLC pathway, but it's cell-specific. Some cell can only has a specific G protein that can only activate this pathway. Other cell can have a specific G protein that can activate the other pathway. And some cells can have both. So some cells can actually activate both pathways. So that's the combination that I was going to talk about later on, but since you ladies are asking. So always keep in mind about cell specificity and pattern of gene expression, which they go hand in hand, right? So this is just still, as much detail as I'm giving you, it's still very general, right? It's still very extremely general. Okay, so like I said, even with cyclic AMP can be a coenzyme for, let's say, 24 enzymes. So then how does the cell know which enzyme is going to be activated? Well, it depends on the cell, right? Not all cells will express all 24 enzymes. Not all cells will express the amount of enzyme at the same level, right? Yes? Yeah, okay. Just want to let you know, just want to see if you understand that everything that we learned so far is like mashed in here. Okay? And that, okay, so cyclic AMP pathway, are we good? Good. Okay. There you go. Depend on cell tissue. I forgot that this slide was here. Tissue specific, cell specific, hormone specific. Specific hormones would activate specific target tissue or target, not the tissue, it's cell, which then each cell would regulate a different effective function, but different effective functions are regulated by different transcription factor, right? So it's not always PKA, it depends on the cell.
So there's multiple cyclic AMP pathways.
I'm sorry?
There's multiple cyclic AMP pathways.
But it
It's
all
just an
all hinges on the cyclic AMP. That's why all these pathways, they're named because of the bottleneck, the one molecule or one enzyme that holds everything together. Right. So yes. Yes. Okay, the phospholipase C pathway. Here's what happens is all the way up until the G protein is the same, right? So signal binding and activating a receptor, which in turn activates a G protein, and activating a G protein means the exchange of a GDP for GTP. I don't need to say that anymore, right? And G protein, depending on the G protein, it can activate another effector enzyme, and in this case it's phospholipase C. What does phospholipase C do? Phospholipase C is an is a peripheral membrane protein. So usually it's not associated with the membrane. When the G protein is active, it actually pulls the phospholipase C toward the membrane and fix it there. Why would it want to pull the phospholipase C to the membrane? Because that's where the substrate is. What is the substrate of phospholipase C? A phospholipid in the plasma membrane, right? Phosphoinositol, phosphatidylinositol. Okay. So when the G protein is active, it's going to again recruit and indirectly because there are a bunch of steps in between. There are how many steps in between? There are like 6 steps in between, which we're not going to look at at all. To indirectly activates phospholipase C. Phospholipase C then would literally grab a hold of a phosphatidylinositol in the plasma membrane and break it in two. Breaks it into DAG, or diacylglycerol, right? And then the other piece is IP3, which is inositol triphosphate, or bis-inositol phosphate. Never mind, just different names for it. Organic chemistry drives me nuts. So basically what this is a DAG is, if you remember, phospholipids is composed of a glycerol, right, a sugar, a glycerol, and it has 2 fatty acids attached to it, and then a bulky hydrophilic head. So the IP3 is basically the bulky hydrophilic head is removed. And then DAG is what remains, which is the glycerol and plus the fatty acid. And so therefore, because of fatty acid, the DAG actually remains attached to the membrane. So far so good? All right. So in this case, you yet have another divergent crosstalk. DAG would lead to another path, and IP3 lead to a different path. Okay? So when we talk about divergent crosstalk, even when you say divergent crosstalk, there are two different ways to view it. One, in terms of divergent crosstalk, let's say at the level of the G protein, then a cell has a choice. It can either go this way or that way. That is divergent crosstalk. However, if you talk about divergent crosstalk at the point of when phosphatidylinositol is broken, then both pathways must occur, right? So divergent crosstalk can either be a choice or it can be a requirement. So in this up here is a choice up here. Here is a requirement. Both DAG and IP3 must be produced for this entire pathway to work. If you somehow negate or cancel out the activity of DHG, then the activity of phospholipase C means absolutely nothing. Okay? So far so good? All right. So you have, again, a divergent crosstalk here. Let's talk about the IP3 first. What does IP3 do? IP3 is you can view it is another example of a second messenger. Okay? And what it does is that it will either get transported to the ER and activate a calcium ion channel in the ER membrane, or sometimes it can actually move and activate the calcium ion channel in the plasma membrane as well. So you have calcium ion channel in multiple membranes. Can be plasma membrane, can be the ER membrane. Okay? Yes, ma'am.
So can it is that reattaching to the
Not reattaching. No, not reattaching. It's just activating the calcium ion channel.
But
So-
it can't reattach.
No. I see what you say. Reattachment occurs where? Or synthesis of phosphatidylinoxidil occurs where? In the ER. Yeah. There you go. Very good. So then, remember, calcium is sequestered, right? Sequestration of calcium is a means to regulate protein function. What does that mean? That means that normally, if calcium isn't in the cytosol, then all of the proteins that require calcium ion as a cofactor will not be active. And when you want those enzymes to be active, then you want calcium to be released into the cytosol. So how do you get calcium ion to be released into the into the cytosol? By the activity of IP3 activating and opening the calcium ion channel. And then sequestration occurred early on. Now you have a movement down the concentration gradient. Now you have calcium in the cytosol. And remember what we said about calcium, that most of the time calcium ions have to be delivered to specific enzymes by specific calcium ion binding protein, right? Yes, sir.
Can you just repeat the function of the calcium sequestration? So after the IP3, so assuming the IP3 gets transported to the ER and activates the calcium ion channel, then what happens?
Right. Then you would have to have calcium ion sequestration before. And if you had sequestration, then calcium ion will have moved down the concentration gradient into the cytosol. What happens if you, for some reason, the calcium ion pump calcium ion back into the ER is broken? Then you don't have calcium ion sequestration, therefore you will not have a means to regulate enzymes in the cytosol that require calcium ion.
So the calcium has to be sequestered. It can't just be in the ER before the second messenger activates.
No, if it's in the ER, then it is sequestered.
Oh.
That's by definition. That is sequestration.
Okay, cool.
Right. Okay. So let's put this all together. So calcium is here. That's released. Yes, sir.
I had a question on the previous slide. Okay, so it does that pathway and it gets to the IP3, right? Is there a second messenger? All right. So how does the cell distinguish from the IP3 binding with the calcium channel on the ER versus the plasma membrane? Is it a specific conformation?
No,
Or-
there's none. It has to do with the particular type of calcium ion channel. Right, so there are there are different calcium ion channels.
Oh, I understand that one, but I'm just saying, like, is the IP3 is it a specific molecule or does it have different conformations or?
IP3 will recognize a certain types of calcium ion channel. So, and when we say that it recognizes certain type, meaning that it does recognize certain conformation.
Yeah, I understand that part. Yeah, yeah, it's different type of IP3. Yes.
No, IP3, no. IP3, by
Yeah,
name,
so-
again, that's why it's
Yeah.
organic chemistry. When you say an organic molecule, that's the only
Yeah.
that's the only version, right? I see what you're saying. No, inositol is really a nucleotide base, right?
Okay.
Nucleotide base, and it has just 3-phosphate attached to it. That's it.
Yeah, that's where I'm getting confused here because I understand that the calcium channels they all have specific conformations, right? So they have different ones in different areas, right? So like the one on the ER, is different from the one on the plasma membrane.
Not necessarily.
Not necessarily?
Is what I'm trying to say.
Okay.
Is that not necessarily? Could be good. They IP3 can actually activate. There are calcium ion channel here that can be recognized by IP3 and be activated by IP3 as well. So the question might be, well, is there a difference between the calcium from out here and in here? And in this case, no.
Yeah.
As long as you have calcium in the cytosol.
Yeah, I'm just
Later
saying,
on,
like,
okay.
like so. How do you? Yeah, how do you
Sorry?
know which one to go to? You know, instead of like accidentally activating both of them.
It could activate both of them. Is what I'm
Oh,
trying to
okay.
say.
It has the same affinity for.
It has the same
Same affinity.
yes, yes.
So there's no control on what you activate then.
No. And you don't have to.
Unless
All
the
you're trying to
calcium
do is get
sequestration.
calcium into the cytosol, it doesn't matter where it's coming from.
Oh, okay, that makes sense.
What Zach just said.
That makes sense. Okay.
What Zach just said. Yeah, that's I'm not saying that you're making it more complicated, but it is more complicated. It actually very simple, is what Zach just said. As long as there's calcium. Now later on when we talk about muscle contraction, where the calcium comes from is really important. But
Okay.
right now it doesn't matter.
Okay.
Right? Now it doesn't matter, and in fact, even though in this picture it shows the ER, but the calcium ion channel in the plasma membrane is also activated.
Okay.
Equal affinity. Really, it has to do with the amount of transport proteins and everything else that is required to transport IP3. For instance, let's say you don't have you have a lot of diamine, but you don't have a lot of myosin.
Okay.
Then chances are that IP3 will get transported toward the ER and not toward the plasma membrane, right? So other things that regulate that.
Okay.
So okay. So
I got you.
That's good. Thank you, Zach. The answer is as simple as that. Okay. So the enzyme, one of the enzymes is activated by that is used that uses a calcium ion as a cofactor is another PK protein kinase C instead of A. Okay, PKC. PKC requires calcium ion, but PKC, the substrate for PKC is actually right by the plasma membrane. So what happened is that in addition to requiring calcium ions, PKC needs to be pulled toward the plasma membrane as well. And PKC, when it's pulled toward the plasma membrane, it acts as a peripheral membrane protein, right? And so how is it pulled or recruited to the plasma membrane? By DAG. So if you don't, this pathway is divergent crosstalk. If you don't have both DAG and IP3, then PKC will never be able to carry out the reaction. So you have to have both. So PKC is recruited. So calcium ion is going to be delivered to the PKC by a particular calcium ion binding protein. It just happens to become modulin, but it doesn't matter, right? As well. And now you have divergent crosstalk, and then it comes back and you have convergent crosstalk here. Because the DAG goes one way, IP3 goes the other way, and then eventually they'll meet back. Both of them are required to activate to get the PKC to catalyze a reaction. You see that? So you have divergent crosstalk and then convergent crosstalk again. See that? And then now PKC is going to carry out a PKC signal transduction pathway. Okay? And then we're not even going to continue with that. So the idea here, the idea of talking about showing you the PLC pathway is multiple fold. Number one, when we talk about divergent crosstalk at the G protein level, then the G protein has choice between cyclic AMP pathway or phospholipase C pathway. When we talk about divergent crosstalk at the level of the PLC, then it is a requirement that both DAG and IP3 are produced. Divergent crosstalk, but then you also have convergent crosstalk. So that's why these two pathways are actually they're great to teach because you see all type of variation in signaling. Okay. And again, you see that everything is tissue specific and cell specific, not only tissue but cell specific and enzyme specific. Different signaling molecule would tell different tissue or cell to perform different function, okay, using the same pathway. And that's, I think LC asked that question earlier, right?
So using this, that's what.
This particular PLC pathway, if you have different signaling molecule, you would activate different receptor in different cell type, and different receptor in different cell type would activate different G protein, which then would activate the same PLC to carry out different functions. And it's because of pattern of gene expression, cell specificity, and enzyme specificity. Continue to ask question. I didn't understand your question, so that's why I can't answer, but that doesn't mean that I don't want you to continue to ask questions for me to clarify. Okay? Again, I'm very slow, so. Yes, sir.
So for this, the divergent aspect, is that both the PLC and the PKC are activated at the same time?
No, divergent crosstalk at that point is that both DAG and IP3 are formed, and each one would go in its own path.
And then it's convergent because
Convergent.
the end.
PKC requires both the activity of IP3 and the activity of DAG. Right. So divergent and converge, and you know, and then there are, let's see, I think like something like 8 more steps before gene expression can occur. And it's people who, scientists, geeks, they're creative. They have names that are just out of this world. One of the enzymes is karma. But we're not going to go into all those details, okay? But I think that by showing the 2 pathways so far, you see multiple big ideas, right? And again, on the exam, I will ask about big ideas using specific examples, details as examples. But you need to understand the bigger idea, okay? And I will ask about the bigger ideas, okay? Yes, ma'am.
Do you want us to know these specific tables?
Oh, no, not yet. You will with liver and pancreas later on, but right now, no. If I have a table, so far you don't have to memorize any table, right? And I don't think you will ever have to memorize any table, because whatever I want you to know will be explained in detail later on. Okay, receptor tyrosine kinase. So a type of what we call enzyme-linked receptor. Okay, so remember there are 4 different types of receptors that we'll talk about. Membrane receptor, G protein link receptor involve 2 different pathways. So here is an enzyme link receptor, also involve 2 different pathways. Okay, this enzyme linked is because the receptor itself is an enzyme. So whereas in the G protein link receptor, the receptor itself is not an enzyme, right? It's linked to an enzyme. So enzyme link receptor, most of the enzyme link receptors, or at least there are different types of enzyme link receptor. Receptor tyrosine kinase is the major type of enzyme link receptor in animals. In plants, it's actually receptor serine threonine kinase. We'll talk about that later. Okay, so in animals, receptor tyrosine kinase constitute the majority of the enzyme link receptors that we know of. And then growth factors, so these perform functions within the paracrine system mainly, but also endocrine system, and mainly paracrine and endocrine system. Okay. I'll have a slide later on, but it's a receptor serine threonine kinase. So there's a whole family of them. If you ever hear about, when people talk about breast cancer, the HER2 receptor is a type of receptor tyrosine kinase, right? So that estrogen, the estrogen receptor is a type of serine receptor tyrosine kinase, And so there's a whole super family of those as well. They look differently, but they have similar function, which is the fact that they do have the tyrosine kinase domain in the cytosolids. Okay, so here's how it works. Here's how, in general, how all receptor tyrosine kinases work, is that you always have two molecules of this receptor. If you only have one, it will never work. So you have two. But normally the two are, two things about it. Number one, the two are not active and they're not even touching each other, right? Number two is that the two are the same molecule. Eventually, they'll form what is called a homodimer versus a heterodimer, right? So they will form a homodimer. So those are the two things about an inactive RTK or receptor tyrosine kinase, is that they are not interacting with each other, and however, they are the same molecule. Then you would have a signaling molecule that would bind and actually not only pull the two receptor molecules together, but cause conformational change of both. So now they are interacting, right? So now you form a receptor dimer, an RTK dimer. Now this RTK dimer is not phosphorylated yet, right? So it's an unphosphorylated RTK dimer. So far so good. The unphosphorylated RTK dimer actually has an enzymatic activity. And then a phosphorylated RTK dimer has a different enzymatic activity. Okay? So we have to talk about them separately. So therefore, dimerization itself is a means, as we talked about it already, right? Dimerization is a means to regulate protein enzyme activity, right? So dimerization of the RTK dimer would then activate the unphosphorylated RTK dimer. The unphosphorylated RTK dimer, once active, would carry out autophosphorylation. Remember, we crossed out that term cross and we replaced it with auto. It's because if we consider this dimer to be the same entity, then that dimer then would phosphorylate itself, right? So the unphosphorylated dimer would carry out autophosphorylation to form a phosphorylated dimer. When people who study this, they would actually refer to this not as just a phosphorylated dimer, RTK dimer, but a hyperphosphorylated RTK dimer, because there are a bunch of different phosphate groups that are attached. And the phosphorylated RTK dimer also has kinase activity, but it catalyzes the phosphorylation of other enzymes, not of itself anymore. So again, the unphosphorylated RTK dimer catalyzes autophosphorylation. The phosphorylated RTK dimer then would phosphorylate and usually activate other enzymes. So far so good? It has yet one more activity that we already talked about, is the fact that this phosphorylated RTK dimer can also act as a scaffolding protein. So what it does is that's why there are multiple phosphate groups, is that the multiple phosphate group would recruit multiple enzymes, right? And each enzyme that is recruited is in turn phosphorylated, but then only enzymes that work within the same, would go toward the same activity would be recruited. So by acting as a scaffolding enzyme, a scaffolding protein, as well as an activating protein, then the phosphorylated RTK dimer can do both at the same time. Recruit all the enzymes that are required, and then boom, 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 activate all the enzymes that would go toward a certain activity, like this. Right, so acts as a scaffolding protein, and then either have these enzymes 1, 2, 3 to 1 activates 2, activate 3 to carry out a certain effective function, or recruit. So that's relay, right? Or recruit different enzymes that can each carry out a different activity. And right here is again divergent crosstalk. This is not divergent crosstalk, right? It's scaffolding,
Yes.
it's hardwiring, but it's not divergent crosstalk because enzyme 1 activate enzyme 2, activate enzyme 3, which activate a downstream event or effector function. Versus this is recruitment, but each of these enzymes would carry out a different activity, and so therefore, scaffolding and then divergent crosstalk. Again, big ideas. And look at this. One of those enzymes is phospholipase C again. So phospholipase C can be activated by either a G protein link receptor or an RTK. And we consider this to be convergent crosstalk. So convergent crosstalk, just like divergent crosstalk, can either be one or the other, right? And a different type of convergent crosstalk is that you have to have both of them. For instance, activation of PKC must have the activity of both DAG and IP3. That's a required basically convergent crosstalk versus activation of PLC is a choice. You can activate PLC by G protein link receptor or by RTK. Yes. I'd be more than happy to repeat anything, to explain anything. These are difficult concepts, I understand that. But how we set up the PowerPoints is that we talk about the concept, but then we repeat it with different examples. So after a while, hopefully it should be secondhand to you by very automatically think about convergent and divergent crosstalk, scaffolding, whether convergent and divergent crosstalk is a choice or is a requirement, and all of that. Hopefully, when you see enough of these examples, that it becomes more secondhand to you. Hopefully. I don't know, but hopefully. Yes, ma'am.
Receptor is divergent. Yes.
Right. Yes. Yes. For instance, PLC, you can either use G protein link receptor or RTK. Okay? Yes, ma'am.
What's the example of the divergent crosstalk in volume example?
Divergent crosstalk is right here. His divergent crosstalk is the phosphorylated, and the RTK is going to be phosphorylated, recruit a number of different enzymes, and each one would activate a different pathway. That's divergent crosstalk.
Okay.
Some of these will come back and meet, and so divergent, convergent, they go in and out. Okay?
So there's just
So
one.
that's one. So we already talked about that. Let's talk about an example of a different pathway that is also activated by a phosphorylated RTK called the RAS-MAP kinase pathway. I know you're tired, but I still have to go through this so that hopefully you understand all of this for the next exam. And again, always in the back of your mind, think that these are just examples. You should understand it in a bigger picture form, right? Don't just nitpick the particular details, right? Don't do that. Think bigger, okay? So the idea is that the phosphorylated RTK is going to activate a monomeric G protein. And RAS is a monomeric G protein right here. RAS right here. Normally RAS is inactive. So the idea is that the receptor will usually not interacting and will never interact with the monomeric G protein, but the activation of the receptor will activate RAS. How does that happen? Okay, again, this is not a G protein linked receptor, but this receptor does activate a G protein. It just happens that it's a monomeric G protein, right? So remember, trimeric G protein has that intrinsic activity where GDP can be exchanged with GTP. Monomeric G protein does not. So in order to carry out the exchange of GDP for GTP, there has to be an enzyme that carry out that, and that is referred to as a GEF, guanidine phosphate exchange factor. So E stands for exchange factor, right? And RAS-GEF is an example of a GEF. So, and RAS-GEF, of course, carry out the exchange of GDP for GTP on RAS. So that's easy to remember. It's also known as SOS, but don't worry about that. Okay. So then, however, RAS is usually a soluble protein. So somehow, remember, enzymes are always recruited to the membrane, and it's recruited to the membrane because that's where the substrate is. And the enzyme is recruited is because the substrate is stuck in the membrane. In this case, the substrate is RAS, and RAS is stuck in the membrane. So therefore, RAS-GEF has to be pulled up. What RAS-GEF is, it cannot be pulled up until it binds to the phosphorylated RTK. Well, a phosphorylated RTK does not have a binding site for RAS-GEF. So then RAS-GEF needs a middleman, an adaptive protein, right? In this case, it's GRB2. It needs an adaptive protein that then GRB2 then would dimerize with RAS-GEF, and then GRB2 has a domain that can recognize a phosphorylated tyrosine residue on the receptor tyrosine kinase, that when the receptor tyrosine kinase is phosphorylated, then our GRB2 will be recruited, and when recruited, it brings RAS-GEF with it, which then put RAS-GEF near the inactive RAS protein, and RAS-GEF then can catalyze the exchange of GDP for GTP, thereby activating RAS. I'll repeat that. Of course, I'll repeat that. Okay. So I'll repeat that by going through this the whole pathway. So signaling molecule bind to the two inactive molecules of receptor RTK, then causing dimerization of the RTK, which then in turn undergo autophosphorylation. The phosphorylated form of the RTK then can recruit a number of enzymes and proteins, including GRB2. GRB2 is recruited. With it, it brings RAS-GEF. The function of RAS-GEF is to catalyze the exchange of GDP for GTP on RAS, thereby activating RAS, and that's where we are. RAS used to be, I think it may have gotten taken over, but used to be for a long, long time the most studied protein. Because if you look at diseases, most people who study diseases find out that there's something wrong with RAS. And so people used to think that RAS has such a varied functions, like hundreds of them. But it turns out that RAS only has one function. So, and we'll talk about that one function. Yes, ma'am?
Are they always bound to each other, or does it have to go undergo
Okay, so they are, you mean GRB2 and RAS-GEF, they are expressed, that's a good question, they are synthesized separately, right? And then they have affinity for each other. They have very high affinity for each other. However, you can regulate this pathway by varying the concentration of both. If you have more of one than the other, then we have fewer of those dimers. If you somehow, let's say, block the expression of one, then RAS will never get activated because GEF is not there. Or if RAS-GEF is there, but GRB2 is not expressed, then RAS will also never be activated. So there are different ways to regulate that. So the idea is that they do have very high affinity for each other, but then you can regulate the amount of each that is expressed. I know that that's more than what you were asking, but those are the things that you have to take into account, right? Not just what, but how much and where. Yes, ma'am. I'm sorry? I'm about to tell you. You're telling me to hurry up, right? Okay. Other questions? My daughter says the same thing, and she's only 3, so I know that I'm in trouble. The downstream signals or effective function is about the same thing, and this is what happens. This right here, you can summarize this right here, this pathway right here, as a MAP kinase cascade. Okay, and you just summarize it as a MAP kinase cascade. So essentially, the active RAS, not RES, but RAS, will activate the MAP kinase cascade. A MAP kinase cascade is when you have a MAP kinase phosphorylating, activating MAP kinase, which in turn phosphorylate and activating MAP kinase. Okay. And then there are, each one of these is a big family, so a member of the MAP kinase is RAF1 for instance, and then member of this is MEK, and member of this is ERK, and I actually know the person who, her claim to fame was to actually discover ERK. So that's my claim to fame is through my friend. I always have to say that. Anyway, so then the whole idea is to activate this whole MAP kinase cascade to get to the MAP kinase here, right? So the whole idea about activating the MAP kinase cascade is to activate MAP kinase. So then if you figure out a way to bypass all of this, and MAP kinase is actually activated, then you don't need any signals. And that's what happens in a lot of cells where you have a mutation, where let's say the ERK or MAP kinase is what we call constitutively phosphorylated. So it's always phosphorylated. Then there's no need for a signaling pathway. There's no need for GRB2. There's no need for RAS. There's no need for any of that, right? But that means that the cell is not well regulated, and that's the stuff where that cancer cells are made of, is that mutation in pathways like that, right? And so MAP kinase will then phosphorylate a number of effector functions. Effector proteins. Each one then leads to a number of different effector functions, including altering protein activities, as well as gene expression. We already talked about one of those. How about regulating the activity of stathmin, right? Right. Okay. For the most part, however, MAP kinase, most of them I know of anyway, actually regulate the activity of transcription factor not all, but most of them regulate the activity of transcription factor that then regulate gene expression. So at the end of the MAP kinase cascade, really most of the time you're talking about regulating gene expression, and regulating protein activity is not as prevalent. But again, this is probably still the most well-studied pathway because it involves so many different things. Okay? And this, because all these are families, you realize that RAS, right? Let's say there are 20 different members of the MAP kinase. Then RAS, one RAS, there aren't too many versions of RAS. There are a few, there are 2 or 3, but not too many versions. So each RAS can actually activate a number of different MAP kinase. So therefore, you're talking about divergent crosstalk right there. And each one of the MAP kinase can activate a number of different MAP kinase. You're not only talking about divergent crosstalk, but possibly amplification as well, right? So these are big ideas. I see you shaking your head. This is how you should study. That's why I highly recommend, maybe later on, in later semesters, I might even require people to study together. Don't try to do this yourself, right? You're going to drive yourself crazy. Talk back and forth, discuss this, and actually it becomes easier once you discuss this, right? Don't try to memorize all of this. It's a lot. Yes, ma'am.
Sorry, could you repeat the big ideas that you just said?
The big idea is that because there are very few. There are, I think, about 3 RAS. I think there are 3 that I can think of, different RAS proteins, right? But there are, let's say, 20 different MAP kinase. That means that each RAS protein can actually activate multiple MEK kinase. So that in itself is divergent crosstalk, right? That's divergent crosstalk. And then that divergent crosstalk continues when each MEK kinase can activate a few other MEK kinase. So that you're actually having divergent crosstalk exponentially. And then in addition to that, each RAS doesn't just activate one MEK kinase, one molecule. It can repeatedly activate MEK kinase, the same molecule, the same type of molecule multiple times. And that is amplification. So you have amplification in each step.
Okay.
So the take-home, an even bigger idea, the take-home message is that if you have a shorter pathway, let's say of one or two reactions, then you have fewer chances of amplification, fewer chances of divergent and convergent crosstalk. If you have a pathway like this that has like 10 different reactions, then at each point you have a chance of having divergent crosstalk, convergent crosstalk, as well as amplification. And I already showed an example of how amplification can occur exponentially with the one photon, right? One photon activating about 500 or so molecules. That in itself is amplification, but then each one of those will activate more, right? So amplification can expand. So again, the simplified big idea is that if you have a longer pathway, then you have more chances of divergent crosstalk, convergent crosstalk, and amplification. Yes, ma'am.
Are each of those pathways related to each other to result in a response? A bigger response?
Yes. Yes. So therefore, you're talking about, that's why a cell must receive multiple signals, right? So we might say there's divergent crosstalk here, but will a cell, let's say from one RAS there are 20 different MEK kinase. Will a cell, once RAS is activated, will it activate all 20 MEK kinase? Probably not, right? So you have other signal nodes would say, okay, go ahead and activate RAS, but the RAS then will only be told to activate 2 of the 20 MEK kinase. So that's why that's then required convergent crosstalk, so that not only is RAS activated, but then the cell has to be told which substrate it will activate in turn, right? Does that answer your question? Yeah, okay. Yes, ma'am.
So you're saying that one RAS molecule can activate more than one of the MAP kinase at the same time?
Not at the same time, not at the simultaneously, but since an enzyme can be reused, then it can activate one molecule of substrate, and then when that goes away, it recruits another one and activate that as well, right? And then, right, okay, that's it. It's a,
What I was gonna say, in this case, it's sequential though, the divergent crosstalk, right?
well, the MAP kinase is sequential.
Yeah.
But if you have, let's say, MEK kinase, let's say you have RAF1 and RAF2. RAF1 might activate MEK1, and RAF2 might activate MEK2. So it's still sequential, but each particular enzyme will have an affinity for very particular substrate. Let's say RAF1 will not have affinity for MEK2. So then the choice would be, is RAS going to activate one molecule of RAF1 and one molecule of RAF2? If so, then one molecule of MEK1 and one molecule of MEK2 will be activated. Versus, is RAS going to activate 2 molecules of RAF1? That in turn then will activate 2 molecules of MEK1, but no MEK2 would be activated.
Right, but each of those would be linear, right?
Each of those will be linear, yes,
like
yes.
The RAF,
Right.
the RAF is only gonna activate
Right,
one
down this way.
and then that MEK's only gonna activate
Right.
one per,
Right. So in this case, the divergent crosstalk is a choice, not a requirement.
Right.
Right. Yes. Whatever you just said, yes. I think somebody else has a hand back here. And I'm going to use as much time as I can, by the way. No more? Okay. Luckily, the tough pathways are done. The tough pathways are done. Okay? So, again, just a reminder that, remember that the RTK can also activate phospholipase C, and it can actually activate the Src kinase family, which is probably the oldest. People know about Src kinase. That's the first kinase that it was discovered. Okay, how that? So about 100 years ago. Okay. And also it has to do with focal adhesion, which we'll come back to that later. Okay. Other enzyme links cascade. So not all enzyme link cascade are themselves enzyme. I mean not enzyme. Not all enzyme linked receptors are themselves enzymes. Some of them are linked to an enzyme. And not all enzyme receptors that are enzyme themselves are RTKs, right? They can be, like I said, receptor serine threonine kinase. Okay, so those are two different ones. There's JAK-STAT, and I don't think we're going to look at that. I believe that we're going to look at the TGF-beta superfamily, because that has something to do with later on when we talk about wound healing. Okay, so be aware of the fact that you do have the receptor tyrosine. You do have enzyme linked receptor that are not themselves enzyme, that they have to be linked to an enzyme. And the example of that is the JAK-STAT pathway. Again, we're not going to discuss that. We're going to discuss the receptor that is a receptor serine threonine kinase. Okay. In this case, you also have to have two molecules of the receptor. However, they are different molecules. So there's a whole family of these receptor serine threonine kinase receptor. That's redundant, receptor kinase. But so when they are bound by a signaling molecule and dimerized, they form a heterodimer.
Okay, so two different ones.
That's important. Later, I'll describe it, and then I'll come back and say why that's important. Okay. So then the heterodimer then also carry out autophosphorylation, so it phosphorylates itself. Okay, and the phosphorylated dimer, receptor kinase receptor serine threonine kinase dimer then would phosphorylate protein called SMAD, either SMAD2 or SMAD3. Okay, in this example. And then when one of the SMADs is phosphorylated, it then can dimerize with another SMAD. So again, forming a heterodimer. Again, very significant. There is a significance between a homodimer versus a heterodimer. Okay? In this case, the heterodimer, SMAD heterodimer, acts as a transcription factor that would bind to a response element to regulate gene expression. Okay?
Oh, okay.
So in the 1 minute I have, so let's talk about the significance of heterodimer versus homodimer. Think about, number one, how quick this pathway is. Because it's so quick, how many different divergent crosstalk, how many different choices, how many different variations do you have? Not much. Right? Not much. So because you don't have a lot of variation in terms of divergent crosstalk, the variation that you do have would be in the particular monomer that you choose. So you would have, instead of the RTK, you just have a homodimer. That's it. You don't have variation. It's just a homodimer RTK, right? In this case, you might have, so if you have a homodimer and let's say the family of RTK, there are 10 members, then you would only have 10 different dimers, right? If you also have 10 members, but if you form a heterodimer, the statistic would be 2 to the 10. That's how many, is that right? Or 10 to the 2? 10 to the 2. No. Yes, 10 to the 2, right. 10 to the 2. So you want to have 100 different variations. Does that make sense? So the 100 different, 10 to the 2, 100 different variations then, each one can activate a different SMAD, right? And then you have a bunch of different SMAD, And then you have different combinations. So at each step, if you have heterodimers, then you can have more variation, which then can get you, can also get to the goal of activating gene expression of a variety of different genes instead of a smaller subset, right? So that's the importance of forming homodimers versus heterodimers.